My name is Thomas Langmann. I'm the head of the Laboratory for Immunology of the Eye at the University Hospital of Cologne. And it's my pleasure to present a talk with the title Retinal Immune Homeostasis and Microglia in the frame of the Foundation Finding Blindness workshop on inflammation in viral gene therapy of the retina. I'd like to start with a little bit of history on the role of phagocytes and macrophages at all. And Mechnikov was the first person in 1883 to really identify these phagocytic cells. He had actually 4 criteria for that phagocytes, which is first, that they are large cells. Second, they have pseudopodia, meaning they can migrate, and that's a third characteristics. And fourth, they can do phagocytosis. Initially, that was found for macrophages that can take up and digest microbes, for example, like bacteria. As a side aspect, Mechnikov, when he was head of the Pasteur Institute later, he was really a fan of probiotics, and that was at that time not so welcome. And therefore, as you see from this cartoon, he, despite his Nobel Prize by identifying these phagocytes, at the end of his life, he was not seen as a best scientist per se. I would come to the phagocytes in the eye. So it's really astonishing that we have phagocytes in the eye nearly everywhere. So we have it in the cornea, on the surface of the eye. And all these things you see here now, we're using reporter mice with green fluorescent protein tagged to macrophage-specific gene expression. And in the cornea, you see this nice image here depicted with C, where these phagocytes make contact to the surface of the eye, like a sort of scanning the surrounding. Then we find macrophages, phagocytes in the iris. We find them in the ciliary body, and we find them in the retina. And this is an image from our lab where we see 2 different images. We see a section, and with the section you can see we have green signals in the so-called plexiform layers, in the nervous fiber layers. And with the whole mount analysis, you can see that the cells are forming a network. I will go to that point. I will focus on that point with the retinal phagocytes that are termed microglia. And Rio Ortega is a Spanish scientist who really was working a lot on working on these cells. And a little bit later than Mechnikov, here in 1933, he was recognizing in the brain that there are these small cells with fine arms, what we call ramification, and they are interspersed between the neurons. And what he recognized is when there is a damage to the brain or an infection, this is what you see on the right drawing, these cells completely change their morphology. So a specific characteristics of microglia is they can do everything what the macrophages can do, plus they have 2 very distinct morphologies. Amoeboid on the right side, we call it amoeboid, like amoebs. They form a network, for example, contacting other cells, what macrophages per se normally do not do. And they seem to communicate with neurons in the nervous system. Some years ago and year by year, there were ARVO special interest groups on retinal microglia. Actually, this SIG on retinal microglia was initiated by Wei Wong, by myself, by He-Ping Su, Florian Sandler, and others, when it appeared that these cells no longer can be regarded as simple bystander cells that are present in the retina, but might have specific functions. And what the colleague and myself found is that there is a very intimate relation between microglia and synapses. This is what you see on the left. Between microglia and Müller cells. They are stained in red here. So communication between micro and macroglia cells. And last but not least, but very interesting also for diseases that are associated to vascular problems, is a very intimate connection with blood vessels. So this might be especially important, for example, for macular degeneration, for a neovascular form, or for diabetic retinopathy. How I envision a microglia cell in the retina is like this, like a spider in its web. And when we look on the microglia here, stained by IBOR1, this is one of the classical phagocyte markers, we recognize them in the flat mount, in the top view, as a network, what I explained before. When we analyze a section, we can see that these green signals are in the plexiform layers. And especially those cells in the outer plexiform layer seem to smell into the photoreceptor layer under normal conditions. So what you see here is a normal healthy mouse and a microglia cell scanning, smelling into that layer. A sort of basic characteristics of the microglia in this normal state is that they sit at their place, they don't move, but they have very active arms of scanning. This is how I envision ameboid microglia cell that has been activated or reactivated. And any kind of analysis we did on histology, for example, using a mouse model for retinitis pigmentosa. Again, you see the flat mount and the section here, that the cells dramatically change their shape. They lose their long arms, they lose their network, and they start to migrate in the outer retina. The same is seen in an experimental model, white light damage as one of the models for dry AMD, or the laser model that is commonly used to mimic the neovascular AMD form. Within the laser spot, we find a lot of ameboid cells that have been attracted from the other parts of the retina. More than 3 years ago, actually, we tried to establish a consensus protocol on the analysis of mouse retinal phagocytes, including microglia cells. And this protocol includes in vivo imaging with transgenic mice that express fluorescent reporters in autofluorescence mode, and then an optimized analysis of histology, sections, flat mounts, and molecular methods like flow cytometry and gene expression. Just as one example, I present some markers. One of the markers here measured in real-time PCR is called AMWeb. Some years ago, we called that protein activated microglia whey acidic protein because it's a small protein found in whey and in milk. And you can see very nicely that this AMWeb transcript and CCL2 as a chemokine are very strongly upregulated. For example, in the retinitis pigmentosa mouse on the top or on the bottom in that light damage model. So using gene expression profiling with specific markers, one can try to measure the activation mode of that cells. I brought this image because I think it's so beautiful that should show you again in a wild type mouse and in a RP mouse model we generated that even at very early stages, and this is seen in this middle image, at very early stages where the outer retina is still pretty normal, the cells start to change their shape. They get fatter, bigger. They still do their projections in the outer retina, but one can already see that something is going on. This is an early state where gene expression, for example, would already indicate an activated state of the microglia. And at late stages, you see it on the right side, the cells started to move, to migrate to the outer retina to eat up the photoreceptors. But work from Wei Wong and from our lab has shown that these cells can actively engulf living cells. So they not only engulf the dead cells, which would be a good cleaning function, but they have a strong bystander effect, killing healthy cells. So for sure, in that state, the microglia are bad guys. Another measurement that can be done is a sort of morphometric quantification, especially in the early stages when microglia start to be activated. This might be important, for example, for the later studies on AAV injections. Even when there's not so much migration, but already the ramification grade, so meaning the volume of the arms and the ramification index are changed. And this can be quantified with a software we have established and published called MotiQ. It's actually a microglia analysis tool. Another tool is RNA-scope, which is a modified fluorescent version of an in situ hybridization. Very often there's a question whether, for example, microglia cells in the retina, and what you see here is immune cells in a laser spot, whether they express certain markers. Actually, often researchers do ELISAs or immunostainings of histology and then claim, for example, that microglia produce certain factors. We believe that the only valuable measurement to show that is RNA-scope in situ hybridization. For example, in this image here, you see the IBA1 positive cells produce VEGF mRNA within the cell, proving that the cells also have a pro-angiogenic vascular damage function. And this is what we published recently in Journal of Neuroinflammation. So that's another tool one can use to determine whether microglial cell responds to a stimulus. We also have a tissue bank here, and what you can see here now is actually an IBA1 staining of a healthy retinal donor microglia on the left side. You see this nicely, beautifully ramified cell. And even at an early stage, AMD, you see that this AMD donor retina network is completely different. This is not a stage where atrophy has happened or neovascularization, but an early AMD stage with a lot of drusen, what we call early or maybe maximum intermediate AMD. Other colleagues, nearly 20 years ago, Gupta et al. Showed the same. They showed in late AMD, geographic atrophy, we see microglia in the outer retina that's actually marked by the arrow, these red cells that have engulfed rhodopsin photoreceptors. And the same has been found by the group in the rare cases where they could do histology of retinitis pigmentosa patients. The same in the damaged retina of an RP patient, the microglia are present, they are fully engulfed. So to us, this says it's not only a mouse-specific thing or a thing you see in experimental models. It has been documented in human patients, in living patients. We will show you later how the concept is, but in post-mortem patients. Microglia as in vivo imaging. I mean, that would be the ultimate goal in a patient to monitor live in vivo the immune response. We are working on this now since some years, especially in the AMD field, using OCT. It's a little bit of debate between groups, but actually I think this is a big scientific discussion that is very valuable. But what we postulate is that things we call hyperreflective foci, or this black dots here, this hyperreflective dots, found at specific site here, for example, in a macular degeneration patient above drusen. You see this drusen images here. And on the bottom slide, even in the neovascular form, when there is leakage in the retina, we believe that these are early signs of clumps of activated microglia. We are trying to go further with that, and we even go to ex vivo analysis of OCT. So you can see it here, that a human eye, and this is a real human eye, you can do ex vivo OCT after the donor has died, and immediately after that you do the OCT image. And what you can see is, and we did a lot of work on histology, that indeed we think we have the proof that these dots close to the drusen that are then later stained with IBA1 and with a microglia marker are very, very similar. We tried to use that hyperreflective foci even for sort of monitoring of disease grade when you count the number of these foci and when you associate it with the early or intermediate AMD stage. And what we found is you find a higher number of hyperreflective foci, let me call them microglia aggregates, with an intermediate AMD stage than with an early stage. This might be important for later, especially with clinical trials where one would like to analyze this hyperreflective foci or immune events, what we speculate in other patients. At the end of my talk, I'd like to briefly introduce the concept of microglia-targeted therapy. Our lab, and I think also other labs, have invested a lot what was initially called immunosuppression or depletion. with this comic here with the tarantula. Just step on the spider and the spider is dead and things are well. This is actually what we did not find. In many, many experiments, we found that killing the microglia did not yield to a better visual function, to a better morphology of the mouse retina. In many cases, it even made it worse. So our concept is what we call immunomodulation. The idea is to let the microglia live, but to transform the cells back to their homeostatic state where they have ramifications, where they eat but don't eat living cells. There are different concepts. In 2008, we published a review on that concept of phagocytes and retinal degeneration as targets for therapy. What we summarized in this cartoon here is that in many different retinal degenerative diseases and neovascular diseases, macular degeneration, inherited retinal diseases, even glaucoma, people find in many, many different papers and models activation of these cells. Therefore, we thought we should be able to document potential targets for this immunomodulation. On this very busy, complex slide that is you can read if you want in the Embo-Molnmed paper, we identified several pathways. In principle, the pathways for the microglia modulation take advantage of previous publications from others and from our group that describe how the microglia get bad, more or less. So they, for example, can be activated by nucleotides, by purines that are released by dying cells. This would be like the purinergic receptor signaling modification, or classical NF-kappa-B signaling, what is known in the immune world very well, or signaling from neurons, from damaged neurons. Our work was also focused on that to identify what are the signals from damaged neurons that signal to the microglia. Can we block that? Another thing is radicals. We know very well that oxygen radicals, reactive oxygen species, are very damaging. If we can block release of that, this could be a potential option. So there exist several ideas, and this is now all matter of analysis in our lab and in other labs as well. A little bit reminding you of the model. So this is a light image model. It's not a genetic model where it takes a long time to mimic the activation of microglia and cell death. In this fast, acute light image model, it takes just a few days. We can nicely monitor the activation of the microglia, their migration, and we can also associate their position in the retina with the activation stage. Another model you might know is a laser model for neovascular AMD, where a laser is used to penetrate Bruch's membrane to mimic the neovascular form. So Bruch's membrane is ruptured, you get a local immune response, you get choroidal neovascularization. The beauty of that model is that you can use any kind of mouse, knock out, knock in, transgene microbiome modification. One can do a systemic injection of a compound, oral gavage, or even intravitreal injection. With that, we just recently published a paper just a few days ago, basically, where we identified one mechanism how this reactive microglia regulate their toxic radical production. And what we identified was a protein called TSPO, translocator protein in the mitochondria. With that paper, we could show that the active microglia need this TSPO protein to produce ROS via an enzyme on the cell surface called NOX1. It's one of the NADPA oxidases. In that paper also, we used a compound called XPD173 that targets the TSPO, modulates TSPO, and with that, we could even change the phenotype of the laser CNV and sort of deactivate the microglia in that system. Last but not least, just one slide of preliminary data that might be important for this group and also for the question how adeno-associated virus might interact with the microglia system. I hope I could show you before that the retinal microglia respond to very, very subtle changes in the retinal environment by the nice communication with other cells. I think I could also show you that degeneration per se can activate them. And we did that study here initially with the aim to express a protein in microglia, an immunomodulatory protein in microglia. But what we saw was the complete opposite. I'd briefly guide you through the slides. On the top left, you see the microglia network uninjected. So that's what you would expect in a normal mouse or in a human person. Nice ramified network, association with the vessels, but no cell clumps, no big problems. The same is nearly true when a subretinal injection of PBS is done. The network is a little bit changed, maybe a little bit shorter arms, but not very problematic. But to our big surprise, when a null vector or expression of a recombinant GFP, or what I call here ORF1 and ORF2, because I don't want to name the proteins we were trying to express here. In each of the situations, when AIV2A combination in that situation here was injected, we see a disturbance of the microglia network. Depending on the expression level and maybe on the titer, I can speak too much about that. We saw differences in changes of morphology. But I think, as I tried to explain you before in the slides, the morphology of microglia is a very big criterion how to judge whether a cell is activated or not. And I think in all of the cases we studied, and the project was also now directed in a completely different direction. Actually, it was directed in a direction of studying the effects of AIV, subretinal and also intravitreal injections on microglia. With that, actually, I'd like to finish, and thank you for your attention, and thank my group. I think you see we are a very international team with a lot of different people, and I'm happy to have these people. Thank you.